Evolution of heat during respiration. Principle. Respiration is a catabolic process which involves the breakdown of food or complex organic molecules into simpler product with the release of energy in the forms of ATP. This process takes place either in the presence of oxygen, that is aerobic respiration, or in its absence, that is anaerobic respiration. The overall mechanism of the aerobic respirations involves the oxidation of carbohydrate and the subsequent production of carbon dioxide and water along with energy. The overall reaction mechanism of aerobic respiration involves the oxidation of carbohydrate, that is the glucose, along with oxygen will give rise to the carbon dioxide, water and ATP, ATP in the forms of heat. To demonstrate the evolution of heat during respiration, we need a three setup A, B and C. In setup A, we have a thermoflask which is half filled with the germinated seeds. In setup A, we need a thermoflask or vacuum flask. I have taken here a vacuum flask which have the fitted thermosensor which shows the accurate temperature inside the vacuum flask. We also need the germinated seeds. Now, how fill the vacuum flask with the germinated seeds? The setup is kept undisturbed for 3 days. Note down the initial temperature. In setup B, we have a thermoflask which is filled with boiled germinated seed. In setup B, we need a vacuum flask along with few boiled germinated seeds. That means we have killed the seeds by boiling it in hot water. Now fill the second setup with boiled germinated seeds and keep undisturbed and observe for the rise in temperature for next 3 days. Now note down the initial temperature. We have setup C which contain the germinated seeds which were killed and surface sterilized with the help of ethyl alcohol. In setup C, we need a vacuum flask, dead seeds, and ethyl alcohol. Now surface sterilize the germinated seeds which were killed by boiling it. Surface sterilization is needed to kill the microorganism which also respire on the dead seeds. And also note down the initial temperature and keep the setup for undisturbed for the next 3 days. In setup A, which contained the germinated seeds, shows rise in temperature for the next day from 25 to 27 in day 1. For day 2, it has risen from 27 to 31 degrees centigrade. For day 3, it has increased from 31 to 35 degrees centigrade. So, in all 3 days, there is a rise in temperature setup B. After incubational period of 1 day, it doesn't show any rise in temperature. The initial and the final temperature reading is the same. But in day 2, it shows some rise in temperature. Even in day 3 also, it shows some rise in temperature. Here, the seeds are boiled and killed. Therefore, the seeds doesn't undergo any cellular respiration. But the rise in temperature indicates some cellular respiration, and the respiration is due to the microorganism which are grow on the dead seeds. In setup C, which contained the germinated seeds which were killed and surface sterilized with the help of ethyl alcohol, all 3 days it doesn't shows any rise in temperature which indicate the absence of cellular respiration, because we have heat killed the germinated seeds and surface sterilized with the ethyl alcohol which kills both the microorganism and germinated seeds. Therefore, there is no any activity of cellular respiration and there is no any rise in temperature at all. Tablet your observations in the tabular column and plot a graph. The graph is plotted in x-axis number of days, in y-axis the rise in temperature in centigrade, and the graphs will come as shown in the figures. The results are tabulated and graph is plotted. From the above graph, we can conclude that germinating seeds placed in the thermoflask shows rise in temperature for few days in the flask A, wherein the flask B, in which seeds have been previously killed by boiling water, the temperature did not rise for about 2 days. The rise of temperature was, however, noticed in flask B after 2 days as microorganism begins to grow and multiply in the dead seed used as a medium in the flask. leading to a rise in temperature due to bacterial respiration. In the flask C, wherein the seeds were killed by immersing them in ethyl alcohol, the temperature did not show any increase, and the alcohol keep the killed seed sterile where microorganisms are unable to grow.